titled What's New in Number 2, Diagnostics for Diarrheal Disease. Dr. Goldfarb is a medical microbiologist and pediatric infectious disease specialist based out of Children's Hospital and BC Women's Hospital and Health Center. Apparently, he also serves as the Associate Head of Department of Pathology and Lab Medicine. Dr. Goldfarb has completed his training and certification in pediatrics, infectious disease, and medical microbiology at the University of Ottawa. And prior to coming to UBC, he had academic appointments at McMaster University and the University of Botswana. His research focuses primarily on novel diagnostic management and prevention strategies for pediatric infectious diseases, particularly in resource-limited settings. So, Dr. Goldfarb, welcome. Very interesting to hear what you have to say, and over to you.
Yeah, thanks so much for that introduction. We'll get started. So just by way of disclosure, I don't have any commercial interests, but there's been various sources of funding I've received for the studies I'm going to be talking about, and I've also borrowed some slides from Dick Garant for this presentation with his permission. So just to explain at the outset, I'm going to be talking about some different technologies that have been used from pre-analytics to analytics in the context of enteric infections, primarily in children, although not all children. And then a lot of it will be focused on areas where diagnostics historically have not been available for enteric infections. So there'll be a lot of discussion around these contexts. That being said, I think a lot of people might be aware there's a big change in our microbiology testing in the province, and we've gone over to these newer techniques, molecular detection. So that just got rolled out within the last couple of months and was involved with developing these GPAC guidelines as well as the cross-validation across the province. So we can certainly, if there's any questions about that, we can discuss that as well at the end. Hopefully there'll be a bit of time. But for the objectives for this presentation is to review relatively briefly the burden of enteric infections in children. I'll talk then in the middle about some of the insights we've had from some of these newer enhanced diagnostic studies, in particular in low resource settings. And then I'll talk about how diagnostics might be able to be used, particularly for children in early childhood diarrheal disease, which is obviously my interest. So the first part, looking at the burden of these enteric infections, you know, on the global scale, and this is all ages, diarrhea, a lot of people don't realize how important diarrhea still remains. There have been some improvements, but it is likely the second leading and communicable cause of death in all comers. But really in kids is where it's the highest burden. This isn't the most recent estimates, but relatively recently, there were over 2,000 children dying a day. And that's more than HIV, AIDS, malaria, and measles combined in terms of cause of death. And it's generally the second leading cause of death in children after pneumonia or lower respiratory tract infection. This is just estimates from IHME looking at the number of deaths by age category. We have been making improvements in actual directly attributable deaths in children, and we've seen improvements in many areas around child health, and there's been a dramatic reduction in under 5 mortality, which we've never seen in history in the last 30 years. And so a lot of that's due to some of the improvements we've seen in diarrheal disease, but still a huge number of children dying each day from this condition. The other thing to be aware about is this vicious cycle and the synergy between malnutrition and particularly enteric infection. And so that concept's not new, and we'll talk about that in a bit more detail. But you know, a child gets an infection. As part of that enteric infection in particular, there's less intake, there's more malabsorption, increased utilization. And so the nutritional status at the end of that infection is worse. That nutritional status being worse then has impacts on their ability to deal with their next infection. And so when they get another infection, it often results in a more severe outcome. And that continues to happen in this vicious cycle. That vicious cycle then may help contribute to a lot of other deaths. So the nutritional status, the reason why we often see much higher rates of death for pneumonia or malaria or measles in low-income settings is because of the poor underlying nutritional status. Here was Bob Black did an estimate that was put in the Lancet some time ago, where pneumonia, it's about half of pneumonia deaths that are due to underlying malnutrition, for example. So what about, though, the kids that survive these frequent episodes of diarrheal disease in childhood? So here's a group of kids in clinic in Botswana that are for all intents and purposes healthy, but we do know that many children in similar settings will experience frequent bouts of diarrhea. Here, anyone who's been involved in pediatric care would recognize this as a growth curve. So this is the 0 standard deviation. A child won't necessarily be at the 0, but we'd expect them to follow a growth curve. This child, this is from not new data, this is studies done in favelas in Latin America in the late 60s, led by Leonardo Mata, where they were following children prospectively and interviewing their care providers to see what symptoms they were having. But this child drops off their growth curve at around 6 months. I don't know in this case, but often that's when weaning may happen from breast milk. And then what you see as associated with that dropping off the growth curve are all of these Ds, and these were diarrheal episodes. And so this can end up being a large proportion of a childhood's experience, is that they're a large proportion of the time they're experiencing diarrheal disease. And so this has been looked at across multiple continents, and early childhood diarrheal episodes are very much associated with stunting, which is a height adjusted for age Z-score of less than -2 standard deviations. And so that's been studied across multiple contexts. And then also looking at the number of diarrheal episodes, whether it's absolute number or longitudinal prevalence. So that's like the absolute number of days that a child has diarrhea in their early infancy. And you can see there's a close association as you have more episodes or more prevalence. And unfortunately, in many settings, you'll have kids more than 10% of their days are experienced as diarrheal disease, which is often the norm, unfortunately, in many settings, and very closely associated with stunting. Now, you'd ask about, well, what does stunting mean? So these are going to be shorter than you'd expect. They're are going to have implications in terms of their neurocognitive development, is where the concerns is, especially in those first 2 years of life, where you have cognitive development relying on heavily on a group of maturational curves, which are kind of demonstrated here that are reflecting cortical development. So there's a lot of development that's still happening after birth, and this is what provides the supporting neuronal structure. Here's image showing you what's happening to for example, the visual cortex dendritic tree during that period. And it can also understand how important nutritional support is for neuronal development, just by looking at the amount of glucose, for example, that's utilized in the brain. In neonates, up to 50% or more of the utilization is occurring in the central nervous system. And so this is why the other big aspect of the burden may, in fact, be human capital. So this is a study by Christopher Epig. It was this graph was adapted into The Economist, but when you looked at IQ, which obviously there are some controversial aspects about looking at IQ across transculturally, but when you look at IQ across 184 countries, the strongest correlate was the degree of infectious disease burden in these societies. That was even when controlled for GDP per capita, education, and temperature. And there was no correlation with nutritional deficiencies when infectious diseases were removed. So further data supporting that we're actually these infections likely have a significant impact on human capital as well that we often don't take into consideration. So hopefully I've highlighted some of the burden, both mortality as well as looking at neurocognitive development. This is a picture I took actually in Croatia a couple of decades ago. You wonder where this car could go with that kind of a burden, but we're going to hopefully later on talk about ways that people are including our work is trying to address some of these burdens. If you're more interested in this, we've done a couple of review papers led by students of mine where we've looked at some of these the data around cognitive burden. So that may be a source if you're interested in looking at that in more detail. So now I'm switching gears now and talking about some of the newer enhanced diagnostic studies that ourselves have done in low-income settings, as well as others have been doing, and particularly the Gates Foundation has funded a number of these. So now we're talking about high burden settings. These are just a graphic representation of where childhood deaths happen. And obviously sub-Saharan Africa and South Asia is where the vast majority now of all deaths are happening, even more so than in 2002. One big study, which was the Global Enteric Multicenter Study, this was funded primarily by the Gates Foundation, and they used what was more conventional microbiology techniques, primarily, particularly for the bacteriology. They did culture to try to recover the organisms, but they also did some molecular for viruses, and then some EIA for some of the protozoan parasites. So the big thing about this study was the size and scope. So it was at the time the largest case control study of an infectious disease in history. I think since COVID, we've probably had larger than that, but they had almost 10,000 cases. And I'd say equally importantly, they had over 13,000 matched community controls. So these are matched for the community, the season, age. And these were done in settings of high burden, so sub-Saharan Africa and South Asia. One of the main initial findings, these were presented actually at PAS in Boston in 2012, was that you had a large proportion of the quote unquote controls. These are children without diarrhea, and the communities had enteric pathogens. And you can see they're actually relatively close in terms of the proportion of kids who had enteric pathogens, even using these more conventional methods. And many of them had 2 or 3 detected. What they were able to do, however, with this analysis is an attributable fraction. So that's looking at the chance of detecting a pathogen in a case, and then in a control, that same pathogen. And that could let you know with a bit more certainty as to how important that pathogen was in diarrheal disease. And the big one wasn't too surprising, was rotavirus. And it had some of the strongest attributable fractions, or it did have the strongest across the kids under 2 years of age. And then another surprising finding was that they also found cryptosporidium was quite important in kids under a year of age. The other aspect of the study is they actually did a 60-day follow-up. And when they looked at the excess mortality, the majority of the deaths, so 2/3 of the deaths, actually happened after that acute illness. So there was a lot happening in after we had done our routine treatments, which in diarrheal disease is generally oral rehydration, as well as zinc in this setting. So there was an excess death that historically often hadn't been measured and was happening in the later period. So some of the main findings: much higher risk of dying. So it was almost 9-fold increased risk of dying, those children who had moderate to severe diarrhea versus community controls. Absolute mortality was as high as 7.5% in some African sites. So that's comparable to, and actually higher than some of the estimates of bacterial meningitis in our setting, comparable to tuberculosis in South Africa in terms of absolute risk of death. So we're seeing in some settings quite a high mortality, and these are even just moderate to severe diarrheal disease. These weren't limited to children admitted with severe diarrhea. An important finding was the pathogens of interest. So those were kind of the leading ones that were found with those conventional methods. Subsequently, there's been now more work done using these broad molecular diagnostic panels. And so with this GEMS study samples, they actually looked at Shigella in particular and did receiver operating characteristic curve analysis in cases versus controls. And using a threshold of 29,000 copies of the IPAH gene, which is present on a plasmid in Shigella, they were finding that probably culture was missing approximately 50% of clinically significant cases. And so that's now been looked at in a number of settings. We looked at this in Botswana, where I had worked for several years, and they actually had done a baseline study using their conventional culture and just stool microscopy, which was available on site. We then evaluated a molecular diagnostic panel. And you can see we went from 85% of samples having nothing detected to just about 7% not having any pathogen, and often finding mixed detection. So much better sense of what was actually going on when we use these broader molecular panels. The Gates Foundation then actually funded a qPCR, so quantitative PCR reanalysis of a large proportion of those GEM samples. And you can see that blue analysis is the reanalysis. Some things didn't change a lot, like rotavirus, because they were using molecular methods before, but other things changed quite a bit. And when you actually looked at Shigella, it became quite important. And in fact, overall, Shigella now had become the most important pathogen with the qPCR analysis. And so that has important implications because there's obviously different interventions if you're going to try to target rotavirus versus going to try to target Shigella. There's another study called the MALI-D study, where this was also funded by the Gates Foundations, and it was multi-center. And they actually collected serial stool samples in children. So this is a different method to try to understand burden of various infections. And so those blue bars are the number of pathogens found when they were doing a surveillance monthly sample. So they're getting monthly samples on kids that were well. If they developed a diarrheal illness during that time, then they got these red samples. And you can see the median pathogen burden, just similar to that case control design, was also quite similar between those two. Of note, they actually found, they did a study in Virginia. This group, the MALI-D group, was based in University of Virginia, and the median number of pathogens in both those without diarrhea as well as those with diarrhea was 0. So just explaining how diarrheal disease is actually quite different in these contexts, and it's a concept of a pathobiome. So these children are, from very early on in life, start to get exposed to enteric pathogens and often have multiple pathogens even by the time they're 6 months of age. And this is this concept of environmental enteropathy. Very early on, there was something called Peace Corps gut that people may have heard about. These were volunteers who went to work in these settings in the 60s and would return with a lot of enteric and gastrointestinal symptoms. They would get endoscopies and they'd find these blunted villi, deepened crypts, other abnormal findings. And we now come to understand that that's actually the norm for many children living in these settings in early childhood. And it's a better understanding of this vicious cycle and how these repeated and chronic exposures to enteric pathogens lead to these long-term effects of stunting and cognitive impacts. So switching gears and talking about another high burden setting in terms of enteric infections in Nunavut, where I'd had worked clinically for over a decade and had collaborated with colleagues there around enteric infections. It's known that many of our northern communities, including in Nunavut, there's much higher burden of gastrointestinal illness. There also are many outbreaks that unfortunately occur in northern communities, and these can be life-threatening infections like 0157, which has been seen on repeated in repeated outbreaks. One of these here, which I highlighted here, occurred while we were doing a surveillance study, and we supported the investigation of that outbreak. There are also other somewhat unique enteric infections like trichinolosis that can present with enteric symptoms. So some of these outbreaks can be life-threatening and can be very challenging to assess the cause of the outbreak, because geography is a real difficult thing. I mean, we deal with this in BC, but in Nunavut, it's even a larger barrier. And so samples, for example, from a Glulik to investigate need to get flown to the regional hub in Iqaluit, and then those are flown down currently to Ottawa before they even get, for example, plated for culture. And you can imagine if you're trying to send a stool sample where we try to get them plated within a couple of hours, ideally with collection, this is often a week or longer before the stool sample even makes it to a microbiology lab for culturing. And so when you look at these reportable infections, they're often very small number, even though we assume there's a high burden. So this is based on national enteric surveillance. In 2012, there were very few reported enteric infections, but we had a hypothesis that there's probably a lot more, and we used this technology, which was newer at the time, open array. It's very small volume PCR reactions, and allowed us to do a fairly broad molecular panel. And so we tested samples that were being submitted for clinical testing at the in Iqaluit, and found that clinical testing very frequently was negative, and if anything was positive, it was bacteria. When we applied this molecular panel, we found majority of the time we found at least one enteric pathogen, and often these were parasites, and the leading one was Cryptosporidium. So 20% of the samples were submitted over that 1 year period were Cryptosporidium. It hadn't been known that Cryptosporidium was even an issue in the north, and labs weren't routinely doing additional stains that we do for looking for Cryptosporidium, like modified acid fast stains. And so when we actually looked at this in Iqaluit, it was amongst the highest reported prevalence compared to almost anywhere in the world if you looked at other similar design studies looking at frequency. And colleagues of ours in Nunavik also started looking more intensely for Cryptosporidium, also even found higher rates. So we had some of the highest rates, of certainly if you compared it to the rest of Canada. So part of that discovery, and then ultimately the availability of easier molecular testing in conjunction with colleagues there, we did implement on-site molecular panel testing in Iqaluit. That's the Kikitani General Hospital, where that was implemented in 2017. And you can see in the just a basic pre-post representation, we went from very tiny fraction of samples having a pathogen reported to now 46% were positive with at least one pathogen. And in particular, if you look at the reportable pathogens, so these are things that are where as labs are legally required to report because there are public health implications, we had a dramatic increase, and outbreaks during this period were able to be, the cause was able to be determined within 2 days, even in island communities that were quite remote. So that's been implemented, and that's one of the most remote hospitals, probably, well, certainly in Canada and likely the world, and they were doing these molecular tests quite earlier on. So we don't know what's going on in Iqaluit in terms of the enteric burden and whether we might be seeing some impacts in terms of nutritional function, particularly in children, but this is one of the concerns and one of the things that is being looked at in the context of Nunavut. So now I'll switch gears and talk about how diagnostics are now being looked at in the context of diarrheal disease in the global context. And here's that same schema of that vicious cycle. So one of the questions is whether we could use antimicrobials to address many of these potentially treatable infections that these children are presenting. So the WHO guidelines have really recommended that we don't give any antibiotics for non-bloody diarrhea. That's been a long-standing. The guidelines haven't really changed much in decades. Like I said, the main interventions have been oral rehydration solution since the 70s. And then more recently, zinc in the last couple of decades has been added. But there's been the recommendation not to use any antibiotics outside of bloody diarrhea. So these are my kids, a couple of my kids, when we were in Mexico a few years ago. The guidelines would say if they develop diarrheal disease, that they would benefit from, in that setting, in a Mexican setting or any other middle or lower income setting, they would get antibiotics, because we have trials in travelers diarrhea that antibiotics will actually improve outcomes. So these kids get antibiotics. Child like this, who's in Southern Africa and may have a 4 to 8% risk of mortality, the guidelines would say do not give any antibiotics if they don't have blood in their stool. When we looked at, in the context in Botswana, we looked at using, this was a Luminex technology that we used to interrogate children admitted with severe gastroenteritis. A third had a treatable pathogen detected, where if they were admitted to hospital, say here, and they had Shigella or Campylobacter, we would treat that in this context. And when you looked at the risk of mortality, it was almost threefold increased risk of mortality if they had one of those treatable pathogens detected, either Campylobacter, Shigella, or enterotoxigenic E. coli. In travelers' diarrhea guidelines, we do recommend the antibiotics as a principal element for the treatment. And these are, whether it's the US CDC or the Canadian guidelines, the UK, Australian guidelines, they all recommend that. But we do know that many of these children who are presenting with their one of their first infections in these settings have pathogens that are similar to what travelers might get. And so this was a recent meta-analysis looking at the risk of mortality. So Shigella, our study was included in that with a larger number of cases, and there were other studies. There's a significantly higher risk of any Shigella detection, regardless of the presence of blood in the stool. And we know the majority of Shigellosis is non-bloody. So there's been some debate, and this was a New York Times article I was interviewed for, where there was some debate, including interviews with the WHO, about whether we need to have improved treatment of diarrheal disease as one of the leading causes of death in children. And so one of the studies that was launched was this large ABCD study, and that's where they're looking at empiric therapy. Obviously, that'd be an easier solution to just give all children antibiotic. There's obviously concerns around AMR. We were wanting to look at the test and treat, and I'll talk about that later. So this ABCD study was initially aimed to have 15,000 children enrolled over sub-Saharan sites in sub-Saharan Africa and South Asia. And their results were just published in their primary results in 2021. They did not find a very large impact overall for all children that were randomized to the empiric antibiotic azithromycin versus the placebo group, but then they have subsequently looked at doing molecular testing of those samples to then see which were the kids that may have actually benefited from treatment with the azithromycin. And so the vast majority of the samples were then interrogated with a multiplex PCR sample with a assay. And you can see a lot of these kids had non-treatable causes. So rotavirus, norovirus, astrovirus, apovirus, etc. And there were, though, a significant proportion, again, perhaps a third, that had bacterial pathogens that would be potentially treatable. So when they actually looked at those who got azithromycin and a treatable pathogen versus didn't get azithromycin, they did find a fairly dramatic, almost a halving of risk of either hospitalization or death. So those who had a likely bacterial etiology, if they got azithromycin, mortality was 4.5%. If they didn't, it was 7.7%, the risk of hospitalization or death. So there were evidence of a benefit, but obviously it's limited to those kids where they have a potentially a treatable pathogen. And so it's really their conclusion is that we need ways to identify which kids will actually benefit for treatment and then aim to offer them treatment. So we went down this test and treat approach. And, the issue with diarrhea, obviously, there's a huge burden of it. There's estimated almost 3 billion episodes. A lot of those are in kids under 5. It's one of the most common reasons people seek care. So a large proportion of people with diarrhea seek care, and many of them get antibiotics already. We know right now only a tiny fraction of those who have diarrhea actually go on and get testing. One of the challenges is around stool collection. And so that was one of the things we want to look at early on. If we're going to have a rapid diagnostic, we know it can often take hours to get a stool sample. And so these are the instructions that are given by labs to tell people to get, all the different containers. They have to go home and get it collected. So we wanted to evaluate these flocked rectal swabs. These are just the swab tip under a scanning electron micrograph compared to the traditional fiber wound swabs. And so we initially compared stool versus flock swabs and found they were comparable. And then we actually compared stool versus the fiber wound swab in this study. And we found that significantly increased sensitivity for Shigella in particular. So the flock swab had 90% sensitivity versus the traditional fiber wound swab was only just over 70%. So we were in discussion about where this might be placed, and this is work we did with the Halteris Associates in doing storyboarding. And so the thought was to look at children being admitted to hospital. Certainly that would be a group that would be severe and where there may be some of the most benefit. And we validated the rectal swab for use in that device over here on the right, which is a BioFire platform, and takes about an hour for the analysis, so fairly rapid. And so you had rapid collection with rapid testing so that you could do a test and treat approach. And we just recently published those results. It was a multi-center study. Unfortunately, we weren't able to reach the sample size. There was a secular trend in a reduction of burden of diarrheal disease, but we were able to demonstrate it was feasible to do in that setting. We did see a trend towards benefit in terms of looking at our risk of recurrent diarrhea for any of those that got to the rapid testing group. We had a 40% reduction, didn't reach statistical significance, but a reduction in recurrent diarrhea. And then when we looked at those with treatable pathogens, again, who were in the rapid diagnostic arm, we did see, again, a trend towards improved nutritional outcome, but we weren't able to reach statistical significance. So we really would need a larger study to be able to look at this in more depth. We are looking at other, perhaps more feasible approaches to diagnostics. This with Mohammed Rehan did some of this work while he was a fellow, where we were using fecal calprotectin, and this is an enzyme that's produced by neutrophils. We use it now in our context for following inflammatory bowel disease, but it also can be seen to be significantly higher in inflammatory infectious causes. So we found, for example, shigellosis and ETEC, there was significantly higher fecal calprotectin levels, and you could use a threshold perhaps as a means to decide which child would benefit from these antibiotics. So the other question is around resistance. We do know many kids with diarrhea get antibiotics. This was a study where they looked at based on the pathogen detection. So many kids who don't need antibiotics are getting them. All these viruses that are here are cases that had received antibiotics and there wasn't likely any benefit. So our thought is that with this targeted approach, you probably could maybe even reduce the overall use of antibiotics. And we also did with colleagues at McMaster, this is just a preprint out, but we did see a no significant increase in azithromycin specific antibiotic resistance based on metagenomic sequencing. So we do have some evidence that we're not actually increasing that at 60 days follow-up. So the goal is to improve growth and development. So a child who would have this kind of a picture, might get their severe bouts of diarrhea diagnosed and lead to a more appropriate growth. So I'll end there, and I'd be happy to take any questions. And so I see there were some maybe questions in the chat, but I didn't see those. So we have a bunch of time I've left for questions if there are any.
Thanks very much, Dr. Goldfarb. Very interesting presentation. There are some comments in the chat. It says that cryptosporidium likely underreported from low and middle income countries due to lack of additional staining required for identification. And then Victor was saying, what were the sources of the cryptosporidium outbreaks?
Right. So maybe just initially Shazia's comment. So that was one of the big findings of that GEM study, is the burden of cryptosporidium. It hadn't been thought about around childhood diarrheal disease as being that important. It was obviously came to the forefront around the HIV pandemic, and how important it was in that setting. But the GEM study, they used EIA, and those had fairly good sensitivity, the immunoassays that they were using. So I think that's how they helped address that. They subsequently did molecular testing and found, cryptosporidium to be similarly important. But yeah, I think historically, people just are not doing stains. Like in Botswana, they were not doing modified acid fast stain looking for crypto, so they was almost never reported. And so I think in many settings, a lot of these infections aren't being reported or detected. And then around for Victor's question, so that had been looked at in a couple of in both Nunavut and Nunavik, and unfortunately, there's never source. There was one study done in Nunavik, and this wasn't related to the outbreak, but it had been done in concurrent time period where they looked at mollusks in that were acquired in Nunavik, that's northern Quebec. And mollusks filter a lot of water, and there's a concern with climate change that when you change the temperature of a coastal water, the mollusks will filter a larger volume of water, and so there may be some concentration happening there. So they found a large proportion of those mollusks did have cryptosporidium detected based on molecular methods. But yeah, the source unfortunately was never found, and we actually had different when we did actually the typing, molecular typing. We had parvum in Nunavut, and in Nunavik it was primarily hominis. So they were different strains as well. Since we've implemented the molecular panel testing, it seems to not been a big issue, but it highlights the important. You can't just do one study and say this is the prevalence of a pathogen in this setting. It's often quite dynamic. So in Nunavut, fortunately, they now have ongoing they have ongoing molecular surveillance, and in Nunavik, colleagues from McGill have helped implement molecular testing for these organisms. So those are now being tested more routinely. But yeah, in the north, I don't think it's that easy. There's no cattle, obviously. There are wild animals. So yeah, often if you look actually the studies around microbiology in the northern settings, there's more studies looking at caribou and other animals in the microbiology than there are in humans. So they've seen cryptosporidium and Giardia before in some other so in for example, dogs been a host where they've seen some of these enteric pathogens, but we don't know, and cryptosporidium hasn't fortunately come back, touch wood, in the numbers we had seen earlier on, but it still is one of these mysteries that we haven't nobody's really discovered what were the sources in those settings.
I see Tyler has a question.
Yeah.
Oh, you're muted, Tyler.
There we go. Sorry about that. Yeah, great talk, David. Seems like a lot of these issues are multifactorial. Like you've got this interplay between nutrition, clean drinking water, all these other issues kind of together, and probably poverty in a lot of these areas, lack of access to resources, healthcare, that kind of thing. But I was curious, you were talking about the microbiome. Do you know has there been people looking at probiotics? Is that something that can help in this area or not? Do you know anything about that?
Yeah, great question. I mean, there's it certainly is multifactorial. Just to speak a bit, though, about the nutritional piece. So a lot of people think it's a lack of access to nutrition. When they have actually done studies with children in many of these settings, their caloric intake and their protein intake is adequate. So it's more the issue around that increased catabolism if you're constantly fighting infections. And they've actually looked at inflammatory markers like CRP and other inflammatory markers, and those kids are chronically inflamed. So the normal fecal calprotectin level, for example, in their stool is much higher than even a child with IBD might have here, just as a baseline. So there is an issue around not just not having access. And then water, there's been some large studies. I didn't go into them, but there have been large studies looking at WASH interventions, water, sanitation, hygiene interventions. So the Gates Foundation, surprise, surprise, also funded those. And these are, again, multi-country, multi-intervention studies that were cluster randomized trials. And they actually didn't find any benefit when you looked at improving access to some clean water, hand hygiene, other hygiene measures. Nutritional interventions where you gave actual protein dense nutrition had some impact in nutritional outcomes, but enteric pathogens, there was no impact. You probably need piped water in all these settings, is what some of the investigators had suggested, but that's like a huge intervention that costs money and resources. But yeah, so I think, and sorry, I was addressing those first 2 points. What was your last question?
I was just asking about probiotics. Is there any
Right, sorry.
potential role for that?
So we evaluated in our intervention, I didn't mention it, it was a multifactorial intervention where we actually also looked at one of the probiotics that's been studied in diarrheal disease. We found, unfortunately, no impact. It was a BioGaia product. And so that actually in that setting didn't have any impact. Now, that doesn't mean to say that it may not be other probiotic interventions or microbiome interventions, but the one that we looked at actually had no impact in that setting. But others have been looking at other probiotics, and obviously, having antimicrobials themselves will have an impact on the microbiome. So people are looking at that in some of these studies where they've done interventions, and we're also aiming or hoping to look at that as well. But that is, it remains to be seen. And from an intervention point of view, what we can do to affect the microbiome.
Thanks, David.
Thanks.
Any other questions for David?
Maybe if I could ask, David, thanks for that presentation. That was really excellent. I'm just wondering if you looked at also susceptibility patterns, especially with Shigellas. As you know, locally, we're experiencing huge numbers of Shigellosis in our under-housed kind of homeless population in downtown East Side, and we're seeing a lot of multi-drug resistance. So we are really here running out of the oral treatment options. In the studies that you've done, has there been any resistance detected, or has that been looked at as well?
Yeah, so we in the study we had done, we were not doing culture there as part of the baseline, though. Their culture and susceptibility had been done on Campylobacter and Shigella isolates, and we had not detected significant azithromycin resistance at the time. This was early in the 2010s. We did do a publication looking at the Campylobacter isolates. It is terrifying because, yeah, when we've had recent Shigella cases here that were travel related at Children's that were resistant to all oral agents and ceftriaxone, and so we had to use carbapenems. That's unfortunately where things may end up going. In sub-Saharan Africa, there's not been that yet. There's not very widespread use of azithromycin, and but that's why it's something we wanted to monitor in terms of its impact to use it, and that's why we think it. Where you use it in a specific setting where there's high risk of mortality, so children with severe disease, and where you're using it in a targeted fashion, the risk of impact in the community. Now, when we looked at that in Campylobacter, we also collected chicken isolates from both broiler chickens as well as free-range chickens, and there was much more resistance in them. That's actually what's driving a lot of the resistance that we're seeing, and many of the genes were spilling over from those isolates into the human isolates. And so I do think it's an important issue, but I don't think it's a reason not to be giving kids a potentially life-saving intervention where there'll be some benefit. I do think we need to preserve it though. And I see Linda as well has her hand up.
Yeah, really nice presentation, Dave. And you guys have done some great work, and appreciate you sharing it today. I just wanted to also add to this discussion that Alex brought up is, we are seeing outbreaks of Shigella, and understanding the epidemiology, like you've demonstrated epidemiology with the pediatric population, hugely important. There's also the downtown east side and various other populations that's it's important to be able to monitor and do surveillance. So all bacterial enterics are reportable through public health. One of the challenges of transitioning to the NAT-based testing panel is the ability to recover organisms. So while you can detect it, unless there's an active process to pull out the positive organisms for culturing and genomics typing, as well as susceptibility testing, we would lose the ability to do the public health response side of things. And so I know that PMAC and the health authorities are very active and engaged, and I can confidently say that in BC, we're aligned with also the federal changes of the case definitions for enteric infections and all that. So I think we're on a good path. The major challenge, though, is what Alex is alluding to, is our ability to collect susceptibility data for surveillance purposes as part of the Shigella identification, genomics typing, and all of that. So we're working closely with public health to see if we can modify that. I just want to say kudos to the work that you're doing, Dave, and thanks for sharing your experiences.
Yeah, thanks, Linda. And also just to mention Nunavut, that was also a concern. It was public health that really drove implementing that testing, because they had their hands tied for a lot of these outbreaks. You could imagine where you have very high rates of reported diarrheal disease. So one of the communities, they asked people when they had an outbreak, how many of them had diarrhea in the last 2 weeks, and 50% of people said they had diarrhea. And so then you're having to send a lot of stool samples. So they really were very interested in having a way to detect and diagnose infections sooner. There, we actually increased the recovery of bacterial pathogens, because when it was identified, the sample would go down to Ottawa with that result saying Campylobacter detected. I mean, it's hard to know for certain to quantify that, but I think a technologist will often, and what we're doing now is actually enhancing our recovery methods when we know what organism we're trying to look for. The problem with stool is that there's literally over 40 pathogens, and you can't do a very intensive workup for all of them, every stool sample, using conventional methods. But when you do this amplification before culture technique, you can then do a much more targeted recovery. And I think this is something on the PMAC side, as you mentioned, we're monitoring. I'm hoping we're going to see similar to what we saw in Nunavut, and actually a higher organism recovery rate for public health purposes, and antimicrobial susceptibility testing. That's really key, as you said. It's quite scary, the amount of resistance we're seeing in Shigella right now.
Any other questions for David? Looks like that's it. Great presentation, David. Obviously generated a lot of interest. Thank you very much for giving your presentation, and enjoy the rest of the day as the snow disappears, I hope.
Yeah. Thanks, everyone.
Bye, everyone.
Bye.